Dear colleagues, thank you for joining this meeting on sheeppox and poultpox. I'm Tsviatko Alexandrov from the European Commission for the Control of Foot and Mouth Disease. And before we start, I would like to give you some technical information as regards the meeting. It will be held both in English and Spanish. And please select the language you would like to listen or to take part. And this can happen clicking and selecting the language as you see down on the right of your screen on this icon for interpretation. Please keep your cameras off all the time due to the large number of participants. You can not speak, you are going to be muted, but you can put questions in the chat box following the presentations that you are going to hear. For the opening, I am very happy to pass the floor to Fabrizio Rosso, the Deputy Executive Secretary of the EuFMD, to welcome you and say the introduction. Over to you, Fabrizio.
Thank you very much, Tsviatko. And I just would like to really welcome you all to this webinar on the current sheeppox emergency. And I would like to thank the colleagues from the EuFMD member nations for trusting us in delivering of this event, this webinar, to their veterinarians and contributing to the preparedness of their veterinary capacity. You probably know that EuFMD has established a program funded by the European Commission that builds on good principle for FMD preparedness and control, and extend them to other transboundary animal diseases, which are similar to FMD. And we particularly consider that the sharing of field experiences among professionals before and during emergency situation is of paramount importance to ensure proper awareness and response. So I really believe that meetings such as this one are vital for improving Europe response capacity to emergency animal disease outbreaks. So we are very glad to see a lot of participants gathered here today, and I hope that you enjoy the discussions and presentation from the different colleagues that have that are available for this meeting. Enjoy the event. Do not forget to follow the EuFMD for any updates. Thank you, and Tsviatko, over to you.
Thank you very much, Fabrizio. Well, the aim of this meeting is to improve the knowledge and capacity for early detection of sheeppox and goatpox. And the discussion will focus particularly on the recognition and diagnosis of the disease. We will have three presentations today. One from the European Reference Laboratory for Capripox viruses from Sciensano on the global situation, the sampling, the laboratory diagnosis and experience from experiments. Then we will have a presentation on the field observations and lessons learned from previous sheeppox and goatpox epidemics in what we have seen in southeastern Europe, and a presentation on the clinical science and epidemiological specificity and the recent experience with the ongoing sheeppox epidemics in Spain. Again, during the presentation, please post any question you have in the chat box that we can address at the end of this meeting. Now I would like to pass the floor to Nick Regge from EURL for the first presentation. Over to you, Nick.
Thank you very much, Tsviatko. First, try to get the presentation on the screen. Is this okay, or should I change?
Change, please.
Change.
Perfect. Thank you.
Perfect. Okay, then I'm going to start off. Hello, good afternoon, everyone. So thank you for having me. I'm Nick de Regge from Sciensano in Belgium, and I'm responsible for the European Reference Laboratory on capripox viruses. So in my presentation, I will try to give you a general introduction on sheeppox. I will also focus a bit on the laboratory diagnostics, and I will try to link this with the results that we obtained during in vivo infection experiments. But let me really start at the really beginning. So sheeppox belongs to the family of the pox viruses, which is a large family which consists of several different genera. And today we'll focus on the genus of the capripox virus. Within this genus, there are three different viruses. You have sheeppox virus, goatpox virus, and also lumpy skin disease virus. And these are all three DNA viruses, which are large, about 150,000 base pairs. And between the three viruses, there's quite some high nucleotide identity. Nevertheless, if you look at phylogenetic analysis, these three viruses cluster in different clades, so they are really three different viruses. What's also important to mention is that these viruses are quite host-specific. So lumpy skin diseases can normally be found in cattle, sheeppox in sheep, and goatpox in goat, although that for sheep and goatpox, this species host specificity is a little bit less strict. So it seems that all different commercial sheep breeds can get infected by sheeppox virus, but the impact of the disease can be different depending on the breed. So breeds from endemic regions show mostly morbidity around 70 to 70%, but the mortality is rather low, around 5 to 10%. This is different from breeds from naive countries, like for example, if European sheep or Australian sheep get infected, the mortality and the morbidity could get as high as 100%, and this certainly in young animals. For the moment, it's not really clear what's the importance of wildlife for sheeppox virus infection. There's a little bit different for goatpox. There are reports that goatpox, for example, can infect coral and red sorrel. So besides the breed, also the viral strain can have an impact on the disease course. Like for example, here on the right, you can see an example of an experiment that we did at Sciensano, where sheep were infected with 4 different sheeppox strains. You can, for example, see in green that for the Moroccan strain, that animals got very severely ill with high clinical scores and that they needed to be euthanized at 9 days post-infection. While for several other strains, they showed important clinical signs between around 7 and 14 days post-infection, but afterwards the animals started to recover, and in the end, they were healthy again. So both breed and viral strain can have an impact. So where can sheep and goatpox be found? This can be more or less seen on this map. So here you can see that most of Africa is endemic for disease. Also the Middle East is endemic, and it can also be found regularly in many Asian countries. The outbreaks in Europe have been very limited the last decades, with just a few outbreaks in Greece and 1 outbreak in Bulgaria. This slide shows more or less the same information, but in blue you can see the countries that have reported an outbreak of sheeppox and goatpox during the last 2 years. And here you can see that several countries from the endemic regions reported outbreaks. But what brings us here together today is that also in September of this year, Spain reported an outbreak. And later on during this webinar, you're going to hear a presentation which will focus more on that. So I think a very important aspect to remember from this webinar is how sheeppox virus is transmitted, because this is very closely related to the control measures that can be taken to stop its spread. So the most important way how sheeppox is transmitted is by direct contact between animals. And then it can be by virus that is present in aerosols, but also a lot of virus is present in saliva and in nasal and conjunctival secretions. You can also find a lot of virus in skin lesions. That's also present in milk, urine, and feces. So these can all transmit the disease. But besides the direct transmission, it's also very important to be aware that there can be an indirect transmission of sheeppox. This can happen through wool, through hides. It can be transmitted between farmers between farms by workers, by veterinarians, by trucks that go from one farm to the other, and so on. And what's also important to remember that when the virus is present in organic material, it can be quite resistant, and stables that were contaminated can also be a source of new infection if new animals are entered in those stables. And something what you might not expect at first sight is that also the environment can play an important role. So the virus can also persist in pastures and can cause the infection of new animals. The role of vectors on indirect transmission is a little bit less known. Probably it can happen, but it's not the major route of transmission. So what makes that there are so many different transmission routes is the fact that the virus is very persistent. So for example, in blood, it can be found for around 1 month. In saliva, nasal swabs, you will detect it for around 2 months. In wool and hair for around 3 months. Then, for example, in scabs, which can contain a lot of virus, the virus can be there and infectious for up to 6 months. And the same is true for pens that where infected animals have been present, and also in pastures, it has been reported the virus to be there for 6 months. So this makes that there is a very large time frame after an infection and after sick animals have been removed that the virus remained present and cause new infections in new animals that enter these stables or use the same pastures. What's good is that the virus is susceptible to the most known disinfectants. It's also susceptible to high and low pH conditions, and by high temperature, you can also inactivate the virus. So the high persistence of the virus makes that biosecurity is very important, that you should think very much on cleaning of trucks on wheels. That's very important to disinfect and clean stables. And that's also very important to think about pastures, because that's not so easy to disinfect. And there, in fact, the best thing is to leave them as long as possible without introducing new animals on these pastures. So what will you normally see after an infection with sheeppox virus? So mostly you will have first an incubation period of 4 or 5 days, but which can take sometimes in some animals 2 to 3 weeks. And afterwards, non-specific clinical signs will develop. The first thing that normally is noticed is a fever, quite a high fever, and you can find some examples on the graphs in the right, where we have done infection studies in sheep, where we tried different viral doses and different inoculation routes. You each time see the onset of a fever around 5 to 7 days post-infection, and you can have fevers around 41 degrees Celsius or even higher. So during the fever, you will also notice swelling of the lymph nodes. You can observe a decreased milk production. You will also see that the animals get less appetite, they get anorexic, and some depression. Then you will have the development of conjunctivitis, rhinitis, with mucopurulent discharges, which can even go afterwards to the development of pneumonia. So after these non-specific clinical signs, you will also have the development of the typical clinical pox symptoms. So this means the development of lesions which start as macula and papilla. And first of all, you will probably see these around the mouth, on the lips. You can see them on the head. What's very important, and often the first place where you can find these lesions is under the tail. And then you can also sometimes observe them between the legs and in some animals over all of the body. So these lesions will then develop further and finally become scabs. Another thing that's important to keep in mind for sheeppox is that you do not only have the lesions at the outside of the animal, but also internal organs show typical pox lesions. So here you can see some pictures where pox lesions, for example, can be found on the lung, in the gastrointestinal tract, but they have also been reported in the kidneys and in the livers. So this is very important because they contribute to the suffering of the animals. So if you have animals with the clear typical pox lesions, it's quite easy to do the diagnosis. And that's also why clinical surveillance is very important, and clinical diagnosis is a good tool. But in some cases, it's not always so easy, because there are some other causes and other diseases which can give similar symptoms if the disease doesn't develop the clear clinical pox lesions. For example, here on the top, you can see 2 animals which were infected with blue tongue virus, and there you also see some nasal discharge and some swelling of the head, which you can sometimes also observe after a sheeppox infection. And the same is true for an infection by the ORF virus, which can cause the ecthyma. Also, these symptoms that you can see here on the bottom can be mistaken for a pox virus infection. So sometimes it's very important to also have a laboratory diagnosis so you are sure which virus you are dealing with. So in the laboratory, we have several different techniques to detect sheeppox virus, and the first couple of techniques relate to the detection of the virus itself. So we have techniques to find the infectious virus, and then we mostly are talking about virus isolation, where you isolate cells in cells. But this is a very laborious test and mostly often done for confirmation. What we do mostly in the lab is detection of viral DNA, and therefore we use several different PCR tests, from which you can see some examples over here. So for example, we have PCRs that detect all different Capripox viruses. Then we have another set of PCRs which allow you to distinguish which Capripox virus is infecting your animal, which will tell you if it's a sheeppox, a goatpox, or for example, Olympic skin disease virus. And then there are other PCRs which are so-called DIVA PCRs, which allow you to tell you if your animal was infected with a field strain, or rather with a vaccine strain, if you know which vaccine was used, of course. And then we have another couple of tests which are more directed to the detection of antibodies. These are the so-called serological tests. And there the reference test is the virus neutralization test, which is, however, very labor intensive. And sometimes, for example, at our lab, it's replaced by the IPMA test. Then we also have an ELISAs for the detection of sheeppox, which allow to look in a large number of samples in a rather cheap way. There is one commercial ELISA, but however, we have to say that this is not completely validated for sheeppox. And then there are other tests like the AGIT test and the EFOT test, but those are not really used very much anymore because often they lack sensitivity and or specificity. What's very important for the diagnostic test is the samples that we receive to perform these tests on. So it will always be good to try to get to samples in which the target is present for a long time and in a high amount. So this means if you want to do serology, it's best to send us blood collected in serum tubes. So normally this works well for serology. If you want to do detection of the virus, then for virus isolation, the best thing is to send us blood samples or material from skin lesions, because this contain a huge amount of virus. Also for PCRs, we can use the same matrices like blood and material from skin lesions. But what's a good alternative is the use of saliva or, for example, nasal swabs, because the virus is present in these samples for a long time in quite a high amount. So in this figure, you can see an example why saliva and nasal swabs can be interesting. It's results that we obtained during in vivo infection experiments. So on the left part of the graph, you can see the results from 7 sheep that we intradermally inoculated ourselves. And there you can see after intradermal inoculation, around day 4, you can detect the virus in most animals in the blood. And by day 7, you can find it in the blood of all the infected animals, and also all the saliva, so the buccal swabs, and also the nasal swabs become positive. But this is after intradermal infection. To these animals, we also added 4 contact sheep, and there those get infected by the natural route of infection. And here you can see that in these contact sheep, the first sheep get already found positive around day 11 or even day 9 in the saliva or in some nasal swabs. And by day 14, we found almost all animals positive in the saliva and in nasal swabs, while in the blood, the first animals only got detected around day 16. So this shows again that these saliva and these nasal swabs can be a good alternative for the early detection of sheeppox virus. Of course, there are some important guidelines for sending samples to the laboratory. First of all, it's important to collect these samples in a safe way for yourself and also for the animals. It's important to think on potential zoonotic agents that can be present in sheep. What's also very important is that you try to collect the samples in an aseptical way so that there is no cross-contamination between samples. And so it's also important to change needles and to change scalpels, for example, if you take samples from different animals. Then it's also for a lab very important that you collect samples in a sufficient number and sufficient volume that we can do all the necessary tests. And once the samples are collected, it's important that you send them as soon as possible to the laboratory, and it's best to do this under cold conditions. And of course, during an outbreak, it's also important to take into account that there can be regulations to send samples because this could be a source of virus transmission. Okay, to finalize my presentation, I have 3 or 4 slides on to introduce a little bit some control measures. And so it's important to keep in mind that the control measures are always aimed to stop direct and indirect transmission routes of the virus. So for example, to stop direct transmission, stamping out can be used. Then it's also important to install movement restriction. This is done by installing protection and surveillance zone. And then you will also, it's very important to do necessary epidemiological investigations. You're going to do active and passive surveillance. And the goal is, of course, to have an early detection of the cases. And then it's also very important that all stakeholders are very aware of this disease. And I think the fact that we are here together today is a good example of this. Then you also want to stop indirect transmission, and for that, biosecurity is very important. You need to think about biosecurity in farms, in milk plants, in slaughterhouses, and all the different actors should be involved. And we speak about visitors, workers, veterinarian, trucks, and so on. Then it's very important to do a proper cleaning and disinfection. And it's also important to keep in mind, as I said in the beginning, that there can be persistence of the virus in the environment. So you should try to avoid common grazing sites. And then as a last control option, there is vaccination. And there exist several commercial vaccines. Those are mostly live attenuated vaccines, and they can be based on different strains of sheeppox. So these vaccines work well. As you can see in the bottom left, there it's an example of an experiment that we did earlier this year where we tested 2 different vaccines and they completely controlled the sheeppox infection, while in the control elements in red, there you see clear clinical disease. If you vaccinate with live attenuated vaccines, also you normally build a quite long-lasting immunity of about 2 years. But the downside is that no DIVA vaccines exist. So once you are vaccinated an animal, you can no longer distinguish if the antibodies that you detect, if they are coming from the vaccination or for an infection. And it has to be said that with the live attenuated vaccines, you can have some side effects. And so the most common side effects that you can observe is a fever upon vaccination. And depending on the vaccine and the batch, you sometimes will see swelling at the inoculation sites, of which you can hear some examples in the picture. So you can see a rather high, big swelling, which even evolves to necrosis at the site of infection, which goes away in the end, but it's not really what you want if you do vaccination. Then there have also been some reports on experimental studies with inactivated vaccines, but I don't think that these inactivated vaccines are for the moment commercially available. So in these studies, they found that these vaccines are working good, that they protect against the sheeppox challenge. And the good thing is that they had less side effects. On the downside, they also don't allow the DIVA approach. And what needs to be kept in mind if you use inactivated vaccines is that you normally need 2 initial doses to get protection, and that you will also need regular boosters because you don't have a long-term immunity. And then I just wanted to mention that other vaccines are in the experimental stage, like recombinant vaccines, multivalent vaccines, but they are definitely not available at the moment. So I think with this, I have come to the end of my presentation, and I will be happy to take questions at the end of the webinar.
Thank you very much, Nick. We have already some questions in the chat for you. I would suggest that we can continue with the next presentation. Dear colleagues, I'm Tsviatko Alexandrov, and on behalf of the EuFMD team, and as someone that was involved in the control of sheeppox epidemics in southeastern Europe since 2010. I will provide you short information on some of the field observations and lessons learned from the epidemics in the region at that time. Sheeppox and goatpox are endemic in Africa, Asia, and Middle East. However, after 2010, they have spread to Israel, Kazakhstan, Kyrgyzstan, Mongolia, and it was further confirmed also in the Russian Federation. Outbreaks were notified in the Balkans neighborhood in Turkey, with more than 1,000 of outbreaks reported since 2010. Although the disease is considered exotic for the European Union, outbreaks were confirmed in Greece and Bulgaria. In Bulgaria, in total, 4 outbreaks in 2013, with more outbreaks in Greece. Last outbreak in the mainland in Northeast territories from 2015, and the last outbreak in Lesbos Island in 2018. While the disease in Bulgaria and Greece was contained and eradicated through stamping out policy, in Turkey, a vaccination strategies was implemented. In the next few slides, we will go through some observations in Bulgaria and in Greece, what we have seen in the field, with pictures from clinical science from the Bulgarian outbreaks. At the beginning, sheeppox clinical signs were not recognized even by experienced veterinarians that worked with the disease in the 90s. The clinical diagnosis at the beginning was ecthyma contagiosa or Plutonk. On this picture, you can see a sheep that was confirmed positive for sheeppox, with what was visible here. Looking from outside, the nasal discharges and generalized hyperemia of nerves and lips in sheep. Again, you can see this in some other diseases in small ruminants. On these pictures, we can see very discrete macules from one side, okay, on the early stage of sheeppox, and on the other side, we can see already the scabs, the old lesions, which shows late detection. But again, I would like to raise to your attention that it's not really easy to recognize, particularly when we speak about the malform. And here, obviously, the disease was lately detected when more animals had shown clinical signs. Sheeppox, malpox lesions, and general lesions on the head on this picture. And again, if you investigate the head, you can see more papules, even when touching the skin. When we opened the mouth, in several cases, we could observe multifocal papular lesions in the upper and lower lips and in the tongue. And when we tested the saliva from this animal, it appeared positive for PCR, and it showed that saliva is an excellent sample for a few tests, and sending samples for PCR testing and confirmation in the lab, even sample better than blood. Some discrete single papules that are not really easy to be detected. Here we have some cutting of the wool to detect these small papules that may suggest the occurrence of sheeppox in these animals. Again, discrete papules, but also scabs in sheep positive for sheeppox. Well, not so generalized lesions as the typical clinical picture. Again, these discrete papules in different parts of the body that you have to look carefully to see and detect. And a node lesion, all discrete lesions in the leg, case where the lesions and clinical signs were missed at the beginning and were detected during this stamping out policy. More visible and more easy to look. It's under the legs, the inner part of the legs, and in this case we can see discrete multiple macules and papules on the axilla, something that's already visible, and it's good when you do the clinical examination in the field to look particularly in these parts of the body. Again, multiple papules on the inner part of the axilla, where they can be typically recognized, and characteristic pox lesions visible around the inguinal area, ventral abdomen, inner thigh, and mammal glands. Again, you just turn the sheep on her back and examine carefully. Characteristic pox papules visible on the mammal glands, and if we go further, the characteristic papules are very visible, what you can see here. Characteristic pox lesions are visible at the base of the tail and in the inguinal area. Recommendation is particularly when you have plenty of animals to examine in a herd, go and look under the tail. This is the place where usually you are going to detect sheep pox lesions, and it can be very easy and fast without losing time to look in more details on the different animals. Characteristic pox lesions again visible on the base of the tail, but here we have the temperature which is above 40 degrees, temperature, the body temperature, and this is indicator for sheep pox now with the fever. Here we have the characteristic generalized lesions visible all over the body, around the inguinal area, ventral abdomen, inner thigh, and mammal glands. Characteristic generalized macules and papules lesions can be visible when cutting the wool, and this is recommended for field test, just to make sure that we speak about generalized infection. And if we go and look closer, we can see the papule and macules visible when cutting the wool, very clear. Post-mortem and during necropsy, characteristic pox nodules distributed throughout all the lung walls are typical. These are just pictures from electron micrograph for an ultra-thin section of papular sheepskin with generalized sheep's pox lesions, many capillary pox virus particles within the cytoplasm of an infected dermal cell. Well, an old method, but again to show you how these viruses look like from the epidemics. And more for histology, sheepskin with generalized sheep's pox lesions, sheep pox cells with pseudophilic inclusion bodies within the cytoplasm, where you can see the arrows. What were the observations? Sheep pox, goat pox has been hampered by the disease underreporting by the farmers. Disease awareness was not good among the farmers at the beginning of the epidemic. Many thought that it was all for blue tongue. And well, maybe this is the reason where there were late detection in some cases, and this contributed to further spread of the disease, and particularly when we speak of villages and common pasturing. You could see different farms affected in same village and area. Clinical examinations and passive farming reporting of clinical signs are at greatest importance for detection of sheep pox and goat pox, and this has to be very clear. These are diseases that are visible and detected by clinical signs. Then, when reported, the diagnosis is easy. And when raising awareness and knowledge in the region, it helped and supported for the early detection of new outbreaks. In the case of large herds, during the clinical examinations, investigations under the tail is quick and reliable method for the detection of the disease. From the Bulgarian experience, saliva appeared to be an excellent sample for PCR testing. The disease was not observed in goats. However, goats should be considered as a risk factor for disease spread, mechanical or by other means. In case of grazing and sharing of pastures, different herds, backyards from same villages were affected. Pastures are a risk for spreading of the disease. and this is some main conclusions, and grazing must be stopped during epidemic. Sheeppox, goatpox was spread indirectly by humans and contaminated materials and/or of transportation of live animals, subclinical or not detected clinical signs, or illegal animal movements. An additional factor that has contributed to the spread of the disease is the partial stamping out of the split affected holdings at the early stage of the epidemics, meaning different holdings of same owner. They were all affected at the end of the day, and it was just delay in the population measures. Jump of the disease to long distances was observed both in Bulgaria and Greece. One farm was affected for second time in Greece after repopulation. However, it is not still clear if the virus had survived there or it was a new introduction. For differential diagnosis, clinical signs of sheeppox, they are highly characteristic. However, in their mild form, they can be confused with parapox virus causing or urticaria from multiple insect bites, blue tongue, and other fast diseases. Well, the pictures on this slide are particularly from these regions. We have picture of FMD, of blue tongue, of ORF, of PPR together with the one from sheeppox, and they all look very similar. That's why it's very important to have the laboratory confirmation as soon as possible. And in case such signs are detected, first to test for foot and mouth disease, sheeppox, pest the petit ruminants, then blue tongue, and then for anything else. In this area, EuFMD together with Bulgaria, Greece, and Turkey is implementing transboundary high-risk area coordinated epidemic surveillance or TRACE program, which includes different components of surveillance activities, but is using again modeling and this camera model to assist achievement and declaration of confidence of disease freedom. From one side is the early detection, from the other side is the confidence of disease freedom, with automized calculation of probability of freedom from fast diseases based on multiple clinical and laboratory data, design data collection and analysis methods. Surveillance data is gathered from Bulgaria, Greece, and Turkey on monthly basis for sheeppox including. Quarterly reports are produced and probability of disease freedom of disease for transboundary animal diseases in trace region, including sheeppox and goatpox, is provided in those reports. And other tools are available and developed by EuFMD to assist members in peacetime and during emergency. The pragmatist, the vaccine prioritization tool that assists vaccine bank management and risk assessors to prioritize antigen vaccines according to the risk and vaccine matching results. The risk monitoring tool, which is a semi-quantity framework to monitor the likelihood of entry of fast diseases, including sheeppox and goatpox, from PR2 countries using information from neighboring countries. From EuFMD member nations with regard to connectivity and different fast diseases pathways effectiveness. The EuFMD, the European scale modeling platform of livestock disease spread and control that simulate transmission within and between countries, now in development for sheeppox and goatpox. Get prepared a toolbox of good practices covering all components of emergency preparedness, and emergency toolbox with open access to online resources as being made available for member countries and other countries to provide open access on online resources. Thank you very much for your kind attention, and we will be happy to address any questions you have. Okay, and from here we will move to Spain. And well, I'm very glad to introduce Germán Cáceres Garrido, who is the head of epidemiology department at the Ministry of Agriculture, Food and Fisheries of Spain, who had prepared a very nice presentation from the Spanish experience. with the current observations on the ongoing epidemic currently. And we are waiting just to start the video. Here it is.
Hello, good morning. I am Germán Cáceres Garrido. I'm head of the epidemiology department of the Ministry of Agriculture in Spain, and I'm trying to show you what we are finding in the current outbreak of sheep and goat pox in Spain. The situation in Spain at the moment, as you know, we detected the disease on the 16th of September. After more than 50 years absent, we knew about the presence of the disease in the north of Africa since a long time ago, but so far we didn't have any incursion of the disease, and this year this change and the unexpected happened, and we detect the disease. At the moment we have 21 outbreaks confirmed by laboratory analysis in the National Reference Laboratory of Regge. 8 of them are in the municipality of Benamurel, Granada, 3 in the municipality of Culler in Granada also, 1 in Baza in Granada too, and 1 in Oria in Almeria, that is a province that is close to Granada, bordering Granada. All of these outbreaks belong to the same cluster. The last one is quite far, a bit farther from the center of this cluster, but it's still in the same cluster. And then we have 8 outbreaks in Villaescusa de Aro in Cuenca, that is this area more in the north, in the center of Spain, that is in the province of Cuenca in Castilla-La Mancha region. Well, these 2 clusters are different in terms of the type of farms that we have there, the type of animals, the type of managing of the herds. In the case of the cluster of Granada and Almeria, we have 12 outbreaks in Granada and 1 in Almeria, as mentioned before. The total census in this area, in these clusters, in these outbreaks, is 2,720 sheep and 123 goats, all of them in holdings for breeding, for production of meat. Plants go then to assembly centers or fattening places for the consumption of meat. In the restricted zone that was created around and now is unified in the whole cluster is 185 holdings, 32 in the protection zone, 3 kilometers radio around the outbreaks, and 153 in the surveillance zone. And in total, there is a census of accessible animals of 48,588 animals in total. In general, they are small and less professionalized farms with outdoor grazing activities that led to the contamination of pasture tracts, what is making control effort and control more difficult in this area. This cluster is still active, detecting new outbreaks in the recent weeks, and we are still investigating and increasing the controls and all the stuff, mainly with the difficulty and the challenge of managing the risks associated to this contamination of pastures in the beginning of the outbreak. In the area of Cuenca, the situation is different. It's 8 outbreaks in the province of Cuenca, in the municipality of Villaescusa de Aro, all of them. Total census in this cluster, in this case, less outbreaks, but a lot more animals, because they are big farms, intensive farms that are, many of them, for the production of milk and some fattening and assembly centers that where we have a lot of animals. We had a lot of animals when the first detection occurred. In total, there are 27,529 sheep in total in these outbreaks. All farms belonging to 2 owners. So we have like 2 epidemiological units, because these owners share veterinarians, personnel, machinery, etc. So we knew from the beginning that there was a connection between all these farms, and so all of them were put under official control and surveillance, and at the end, all of them, little by little, have been affected by the disease, and they have been confirmed, and they have been stamped out already. So we had a total, the restriction zone of 17 farms with a total census of 41,739 animals. And as I mentioned, there are intensive farms, assembly centers, fattening units, milk breeders, so no grazing activities outside the farm, which is very important because that is one of the reasons that we knew that this area could be easily controlled and put under control, the spread of the disease. So the cluster at the moment is stable. So this is a summary table with all the information related to the total outbreaks, 21 that we have detected in Spain so far until today. So the clinical prevalence, the average is 4.69%, maximum 26, outbreak 13, minimum 0.03, ID 15. Of course, older lesions, we've seen that older lesions in herds with higher clinical prevalence, so the disease had more time to evolve and produce these kind of lesions. Higher transmission risk from necrotic lesion and scabs, as you can imagine, when the animals develop the scabs and the lesions, the productive lesions, these scabs fall to the floor, and the virus can survive there for many months. So this has happened in 3 of the outbreaks, in outbreaks 1, 13, and 18. So close monitoring of the places where these animals were grazing in the previous weeks has been very important, and has been tried to put in control little by little, because it's not easy, because these are very small farms that sometimes depends on this. A subsidy has been produced for the farmers, provided to the farmers to allow them to feed the animals indoor, and now is becoming better in this regard, the situation in the area. So in the first moment, as you see in the table, samples and results, we prioritize, the sampling, the rapidity, the diagnosis, and the confirmation of the disease. So we then take many, in the case of symptoms of clear clinical signs, we took just a few animals for confirmation purpose and for the rapid control of the disease in these outbreaks. Now we are ongoing a study where we are trying to take from the new appearing outbreaks a complete statistical significant sampling strategy, and we hope that we will have a lot of information that will be very, very important to understand how the disease behave in the affected farms. So the first isolate was sequenced to determine that it was a field strain, and it was a sheep goat pox virus, and it was a field strain. And then the communication was when this field strain was result was confirmed on the 19th, and samples were immediately sent to the Sciensano, to the EU reference laboratory for sequencing. The information coming from there, from sequencing activities, has been that is related to isolate circulating in Morocco in 2014, Egypt in 2018, and Turkey in 1970. So it's not very clarifying, as you can see, and no clear conclusion about the origin of the virus sequencing. So if we look at the clinical findings that how the disease is described in the OMSA, in the organization on the World Health Organization for Animal Health, the manual says that the incubation period 4 to 14 days. In the case of Spain, the field strain in Spain seems to have a long incubation period, so we would be looking at the highest range of this incubation period, 14 days. We think that is more plausible here. And sheep and goat pox are characterized by disseminated cutaneous nodules, up to 100% mortality in fluid. This is what OMSA said in the manual, but in Spain we haven't seen these high mortality rates in any of the outbreaks. It is true that most of them have been detected in early stages, so not giving time to to increase the mortality, so higher. Goats not seriously affected, they seem far less susceptible to both infection and clinical signs. Another thing that is in the OMSA manual is some breeds of European sheep may die of acute infection before the development of skin lesions. In Spain, we haven't seen this. So we haven't seen any acute case of sudden mortality. May have happened in few animals in the beginning. In any case, nothing very impressive for a farmer would make a challenge the passive detection of the disease by the farmer and private veterinarians. So they have to be close look on the animals, close monitoring of the animals to be able to detect. In all the times in Spain, the clinical presentation and evolution observed in affected animals have been typical. It's described in OMSA manual. Okay. So we are showing here some of the symptoms of clinical findings that we have found in the outbreaks in Spain. And in this case, these are initial stage of lesions, skin lesions, reddish macule, papules, 14 days incubation plus 2-5 days until lesion development. So most likely affected area, easy to check, and useful for passive surveillance under the tail. This is typical, the presentation of clinical signs under the tail of the animal, so that making easier, to the inspection of the animals daily. Within 24 hours of the appearance of generalized papules, affected animals develop rhinitis, conjunctivitis, and enlargement of superficial lymph nodes, particularly because they are easily accessible. The prescapular lymph nodes would be inspected in animals very easily. Then the disease or the clinical lesions evolve in papules into a necrotic lesion and scabs, so more infective from the point of view of these grazing areas and in the farm, in the manure, etc. The scabs are able to have the virus be able for months, so 14 days incubation plus 2-5 days until initial lesions plus 5-10 days to develop the scabs. So we are looking in this kind of clinical findings. At least the disease has been in the farm for at least 1 month, and that is very important for epidemiological investigation, of course. So some relevant information about the epidemiology of the sheep and goat pox virus in Spain is a non-explosive presentation, that's for sure. Few animals infected from grazing fields through incubation period, 14 days, and then slow intra-herd transmission, more animals through incubation period, 14 days, so clinically evidence after 1 month after first infection. So this is very important for epidemiological investigation, and this is what we are seeing in Spain at the moment. Mortality is not very high, and it's progressive in the herd, so it's not very evident. The indirect transmission, main challenge, as I mentioned, is the contamination of common pastures and other grazing fields and tracks. High stability of the virus in the environment, mainly when animals have scabs and productive lesions, as I mentioned before. Other indirect paths, of course, possible. This is a very transmissible disease, vehicle, personal, and vets, vomits. No important role of flying mechanical vectors, and goats are not very seriously affected by the infection and by the clinical disease. The possible origin of the virus is unknown at the moment. Investigation is still ongoing. Field strain phylogenetically related to isolates from Morocco, Turkey, and Egypt, in a very long time period, so this is not very clarifying, of course. Most plausible is the workers with dirty clothes from endemic North African countries who came to Spain and work in sheep farm. This is the most plausible theory that we have at the moment, but we haven't found any clear evidence of this anyway. Other possible pathways, implausibility or illegal direct sale of animals around the feast of the Muslim countries, the 10th of July, to North African people who could have gone inside the farms to choose the animals, for a feast. And of course, the vehicles not properly cleaned and disinfected. And this is very unlikely because we have a very, very important and very thoughtfully applied protocol for these vehicles that are going to the North African country and then coming back to Spain through a specific protocol in the border inspection points because of the risk of foot-and-mouth disease that, as you know, has been around for a long time now. Conclusions: we have at the moment 21 confirmed outbreaks so far in 2 clusters, Cuenca and Granada. Granada, of course, include another outbreak that is quite a bit farther from the center of this cluster, that is in Almeria, but still related to the same risk factors that we detected in the cluster in Granada. They are epidemiologically linked, these two clusters, because to each other by animal movement that we knew about in the beginning during the incubation period, in the high risk period, so before first detection. So we had a clear idea that this area could have been, or Cuenca, cluster in Cuenca could have been infected from the beginning, because we detected movement from Granada to Cuenca in those days. No risk movement identified to other member states in the high risk period. The cluster of Cuenca is stable at the moment without new outbreaks. We hope to leave the restriction zone very soon. Of course, we know that in this area there is no risk for pasture contamination, because they are intensive farms and the animals didn't graze outdoor. So that is making easier the management of the disease in this area. The cluster of Granada on its side is still active, with new outbreaks every few days, probably associated to a sporadic non-compliance with this ban on outdoor grazing. This has been very difficult to apply, because there are small farms, very remote. We are in coordination with the police and giving a subsidy to the farmers to try to avoid these animals from coming outside the farms, so they can be fed inside. And of course, we are doing a lot of awareness campaigns in this area with these farmers to try to make them understand the importance of keeping the animals indoors to avoid any further risk in this sense. The origin of the virus remains unknown. Most plausible hypothesis is, as I mentioned, the work is coming into work temporarily into these sheep farms in this area, but we don't have clear evidence yet about this. So the investigation keep on going. The typical clinical signs which become evidence in terms of number of animals affected after 3 or 4 weeks after first infection in the herd. So it's very important, a close and direct observation of the animals and immediate communication to the official veterinary services of any hint of disease. So enhanced passive surveillance is key in this sense. One of the main challenges is the contamination of common pastures and grazing fields, where the virus can survive for months. In the lab, it has been demonstrated that the virus can be alive or can be infective in this condition for 6 months. And so we are looking at how to make sure that these pasture and tracks are not they don't have viable virus before lifting the restrictions in these areas. It's not easy, but we are trying to prolong the time in which these areas will be restricted until probably the beginning of next year, and we will look at some kind of procedure to make sure that there is no further risk of infection of new outbreaks. Biosecurity is of paramount importance of all levels: farm, vehicles, and field teams. And these are the conclusions from my side. So with this, I finish my presentation. Thank you very much, and we'll have the opportunity to discuss and answer any question that you have or comments during the workshop. Thank you very much.
Thank you very much, Germán. Now we do have some questions in the chat. I would suggest that we can go through a few of them before closing the meeting. And the first one that I would like to put to your attention, as we are now on the topic of Spain. A question: is mixed goat and sheep production sites are both well, in case of mixed goats and sheep production sites, are both species caught? Maybe Germán, if you can try to provide an answer, and I can follow.
Sí, bueno, hablo en castellano, creo que estoy bien.
I'll speak Spanish, because I do believe that you can hear me on the Spanish channel. So first of all, I didn't say it during my presentation, but I wanted to thank the official veterinary services in Andalusia for all of the magnificent work that they're doing. And right now, at an outbreak, we're stamping out both sheep as well as goat. I know that goat don't have the same clinical signs, and the diagnosis is a bit more complicated. It seems that they're less susceptible to infection, but stamping out is for both species right now.
Thank you very much. And well, for the case, what we've seen in Southeast Europe in the past, so both species, they were stamped out due to the risk of contact, although we haven't seen goatpox in the field. Another question, if we can comment on the survival of the virus for years in dry crusts at room temperatures and participation in the transmission of vector insects such as stable flies. Maybe this is a question addressed maybe the European Reference Laboratory.
Yes, maybe I can try to answer for as far there's evidence on that. So survival of scabs, what we can say from literature is that there are a few reports that really mention that scabs contain a lot of virus and it can remain infectious for a long time. But the data that I've seen, they tell about up to 6 months. I don't know if somebody has looked longer into that. And then the transmission by vectors. Yeah, it seems, and it has also been described in literature, that sheeppox can be transmitted in a mechanical way by vectors, but that's not the main route of transmission. There you already have the direct and the indirect contact. Like for other capripox virus, like lumpy skin disease virus, there it's really clear that's a vector-borne disease, but this is not the case for sheeppox virus. You cannot exclude that they can play a minor role.
And then a question that just appeared, what could be done with pastures?
Yeah, that's a difficult one to answer. What you want to do is to leave them as long as possible without new sheep entering on them. I think maybe Germán can tell more about that maybe they can be used for another purpose, to grow vegetables or something like that before sheep are entered on that. But maybe Germán has a better idea.
En esa zona.
Well, in this area, we do know that it is quite desertic area in winter, and rain is always bad for this, but it's also places where there is much culture and harvest, and they're usually being used for planting and planting all sorts of winter vegetables or cereals. And the problem that we have is that once you lift the restrictions, animals can go through again and they could infect themselves again, and we could have new outbreaks. So these things have to be evaluated. We have to do it in coordination with the official veterinary services in Andalusia, because they're the ones who are on the field and know the situation. But we have to stabilize things so that we don't have new outbreaks, and then we will see what to do with pastures that will have to be managed carefully so that we can avoid for the disease to appear again, because it would be terrible, wouldn't it? I don't really have a clear answer about a date or so on.
Thank you, Germán. Thank you very much. And it's really complicated question. Well, again, maybe a question on the maintenance strategy by which the virus maintains, and what about the role of wildlife, of wild animals?
Well, as I said, there is in literature, very little data on that, if wildlife get infected and if they have a long persistent infection. So I cannot say very much about it. So as I mentioned also for goatpox, there are some recent data that show that there are some wildlife species that get infected. But I think, yeah, as we mentioned, the virus is so stable in scabs, in the environment, in organic matter, that's a very important way of transmission. And that's the one that we need to take into account and try to control.
Okay, thank you very much, Nick. And well, just last question that was also raised during the EuFMD open session. Do shear trimming machines used in wool sheep represent a risk for virus transmission?
Yes, indeed, we have had that question before, and now we have discussed on that, and it seems indeed very important because we know the virus can be at the lesions. If you go with a trimming machine over them, your trimming machine can get infected. If you take it to the next farm and you share sheep, of course, it's a way of indirect transmission. So it's very important, and it's not only the trimming machine; it's also the worker who is doing the trimming. That's very important, and that carefully should be managed, and that biosecurity is very important going from farm to farm.
Thank you very much. And well, I would suggest that we can shortly close the meeting, and for the rest questions we can reply by email. I think the ones that are most interested about the origin of the introduction of virus to Spain, it was already addressed in the presentation by Germán. From another question, yes, we will provide a recording both in English and Spanish after we close this event, and it will be available to everybody. And I would like to thank you all that stayed here with us and that listened to these talks. Special thanks to Nick and Germán for their presentations, but also to Chris de Klerk, who supported us a lot in the preparation of this meeting, but not only. Really special thanks to the European Commission for their support. And not at the end, I would like to highly acknowledge the work by the EuFMD team, Rodrigo, Charlotte, and particularly Sylvia Epps and Filippo Padua, who really made this event possible with so many participants around. And I am sure it was not easy for them. Well, thank you very much again, and looking forward to seeing you soon again.